So my name is Barbara Blanco, as I was introduced. I'm part of the plant sciences department, and my research is basically on fruit biology and quality. I generally work with two different crops. Tomatoes are my main one, but I also work with strawberries. And as we are going to discuss in a little bit, I wanted to bring up what's a fruit and what's a vegetable, because this is quite a debate for consumers, but for us biologists, we see fruits that sometimes are called vegetables. So I put just this Venn diagram for you guys here, and you see, well, what we mostly will talk about in this workshop is about what we call fruit canonically, so what we eat that are sweet and are all that on that side of the screen. And then we have other type of fruit, which we have in the middle, avocados. We will talk about it during this workshop, and maybe some tomato will be discussed, but there's other ones that are not going to be discussed because we don't eat them in the ripe stage, and I will talk to you about that. So what's a fruit? So we can call a fruit is basically the organ of a plant that bears seeds, and these seeds are generally used to generate another plant. We know that there's some fruits that do not really bear seed because we have bred for them, because the consumers like them without seeds, but they are still fruit. Okay, so when we were talking about fruit, and we think about the development of a fruit since it starts in the plant, so we have an initiation point until death. So here when we work with fruit that are ripe and are dying and have been harvested, and if you are retailers or you work with post-harvest supply chain, you are trying to avoid this to happen, death, right? So you have all this period in which the fruit develop in the plant, the growth, and then you reach maturations. Sometimes you harvest your fruit in this step, right? When the fruit is mature, and then you condition it or you ripen it basically in your warehouses or storage facilities. Or sometimes you also harvest the fruit when it's already ripe. So it depends on the market, and it depends on the purpose of the fruit, and also on the biology of the fruit. Some fruit cannot be harvested mature because they will never ripe outside. And some of them, you might like to harvest them when they are closer to this stage. So when I was saying there's fruit you don't never want mature, we won't discuss them, but cucumber, squash, you don't want those to ripen, right? So they're harvested early on. So basically, we'll discuss most of these type of fruit that you would like to ripen and see those quality attributes showing up. So ethylene and plant hormones. So plant hormones are basically, you can call them growth regulators, and they are messengers. They're chemical messengers that are produced by the plant. We also can produce them artificially and basically spray them or apply them to plants to get an effect. Hormones interact with receptors in the cells of the fruit or the plant, and they can trigger a signal, and that's basically what it will end up as a response. As in animals, plant hormones are basically capable to induce a response in another part of the plant. So for instance, it could be that ethylene is being produced in one side of the plant, but the organ that is responding is in other place of the plant. And what we are going to discuss most in depth is that ethylene is a gas. So it's a small hydrocarbon gas, and because it's a gas, it's really easy to distribute across a room, across space. And we know that it is the most common ripening hormone. We'll discuss this a little bit in the context of climacteric and non-climacteric fruit, but all plants produce ethylene, and although the rate of the production, as I mentioned here, will depend on the type of the tissue and also the developmental stage of the organ. An immature fruit is likely not to produce that much ethylene or is not capable to respond to that ethylene, whereas a ripe fruit will produce a lot of ethylene and will be more likely to respond to that ethylene. So there's a time in which the organ, in this case the fruit, will be capable to respond to that signal. If you apply ethylene when the fruit is too young or too immature, hasn't reached that physiological maturity, you will never get an effect. So you have to understand what's going on in that fruit. Is that the right stage? You need to understand what's the process, the biology a bit, so you can be able to basically adapt your strategies to ripen the fruit. So one thing that is also important is that as a hormone, ethylene can be perceived at really low concentrations. We basically use a lot of ethylene when we are trying to ripen fruit. We overdo it a bit because we want to make sure that fruit gets ripened. But actually, ethylene can be perceived really low, as I mentioned here, about 0.1 parts per million. So if you are a person working with fruit, you are familiar with ethylene. But ethylene as a plant hormone has a bunch of different functions. And these are important because sometimes you don't just deal with fruit. You might have if you're a shipper or packer, you might have different other commodities that are not fruit in your facility. And ethylene can cause effects in those commodities as well. So you need to understand that ethylene has different functions throughout the development of plants, and also different tissues may respond differently to ethylene. So here I put in just a few of those common functions of ethylene, and I highlighted two of them. One of them, of course, fruit ripening, as we're mentioning, but also ethylene has a role in defense responses. So it depends on the dose of ethylene. You might also prevent some decay or disease in the crop. Now, if you are using an overripe fruit and you have ethylene, might cause the opposite effect. So it depends as well on the dose that you are using. So don't frown your face for me here. This is just an example of the ethylene biosynthetic pathway, and I wanted to put it out there because maybe some of you would like to recognize that as many metabolic pathways in plants, it's a complex pathway. You have the starting point of this pathway is methionine, an amino acid, and it's cycled through this Yang cycle. And I brought it to you because this was discovered here at UC Davis by Professor Changfa Ya, and he basically uncovered this part of the metabolic or biosynthetic pathway of ethylene. But what we need to know in our case is mostly these steps in the pathway which are pretty critical. And why are they critical? Because they are points of management. So let's remove all this complicated graph and let's make it a little bit more simple when we talk about step 1, 2, and 3. So I put it here for you. So we start with methionine, which is basically the precursor, and then you have 3 enzymes that work to produce ethylene: SAM synthetase, AC synthase, and ACC oxidase. And here, in some cases, you can control the rate of ripening or accelerate the rate of ripening if you are able to induce conditions that are favorable or inhibitory to these enzymes. So these basically produce ethylene, but ethylene by itself will not cause any effect. It needs to be perceived by the organ, as I said. So first of all, you have perception. So the organ, the cells in that fruit are able to detect ethylene using receptors. And then that signal, after the receptors recognize ethylene, will cause a response cascade, will happen, a lot of signals will happen inside the cell, and then you will have an effect, which can be seen at the cellular, but also at the level of the fruit. For instance, you get ripening. So you need all this process to happen in case you want to ripen a fruit. So you need the biosynthesis, and you need the perception as well. So now, that being said, let's go into more practical points. For instance, can we consider ethylene to be always a friend, or is it a foe? As I said, it depends. In our case, in this workshop, we will treat it more like a friend, because we needed it to ripen fruit. But sometimes you need to avoid that ripening to happen so fast. And also you don't want that gas to affect other crops that you might have in your warehouse or if you're a retailer in your store. So ethylene can promote beneficial effects if it is controlled and applied correctly, but it also can cause major post-harvest losses. So positive things, of course, ripening, like banana ripening. You can have also citrus degreening. But if you get some vegetables exposed to ethylene, you may get things like potato sprouting. That is something that you really don't want. Or in lettuce or in other leafy greens, you may get some spotting like rosette spotting. So why some crops are more, let's say, sensitive or non-sensitive to ethylene? Well, it depends on their biology itself and also their developmental stage, how ripened or how mature they are. I put together this for you here so you can have an idea of different post-harvest commodities and how sensitive or how high producers of ethylene they are. The ones in light gray are not really fruit, so that's why I kind of uncovered them for you, but you might be interested about them. So here you have ethylene producers. Apples are really high producers of ethylene. And you can go all the way down here into other commodities that produce really low ethylene, such as strawberries and some of the citrus like tangerines. Now, if you go into sensitivity, cucumbers, which are vegetables yet fruit, they are really sensitive to ethylene. So if you put together apples and cucumbers, you might not get the really good effect, right? Your cucumbers will get affected, and you don't want ethylene to you don't want them to be exposed to ethylene. And some of them are less sensitive to ethylene. However, they will still respond to it. And there's some that do not really produce or are not sensitive at all to ethylene, such as those ones here. So that means that maybe these are more compatible to high ethylene producers if you're putting them on the same shipment. So that's an example of different fruits and vegetables. Now, as I was saying, what really influences that a crop or a commodity is a high producer of ethylene? So of course, the genetics. Different cultivars may have differences in their production of ethylene. Their developmental stage, as I said, immature versus mature. Of course, temperature, right? If you have cold temperature, you reduce the metabolism of the commodity, therefore you don't have that much ethylene. The oxygen level. So I told you that pay attention to those enzymes in the biosynthetic pathway. So actually, the ACC oxidase requires oxygen to act. So if you reduce oxygen, you basically inhibit part of that pathway, and you don't get that much ethylene. So you need to understand that maybe you want to reduce oxygen level so you don't get ethylene production, or maybe you want to increase oxygen level so you can use it in basically in a ripening room. So you need to understand that dynamic on the gases that are present when the fruit is in your warehouse or in your ripening room. The same for CO2. So if you have high levels of CO2, you might inhibit the perception of ethylene. If you expose the fruit to exogenous ethylene, you might have also a difference in the production rate. And if you have stress, that means your fruit is bruised or starting to decay, ethylene will start going up. Because ethylene is a response to stress in plants. So as soon as you have a wound, you have a bruise, or you have a pathogen there, you will have increased ethylene. So as I was saying, where does ethylene come from? So could be from a biological source. Your normal ripening fruit will produce ethylene. Or as I said, your decomposing or wounded produce will produce ethylene. For instance, if you have an incomplete combustion in engines, even a car exhaust can produce ethylene. Your forklifts, if they are they are not electric, they might be producing ethylene. If you have propane power equipment, there could be some ethylene produced. If somebody smokes from the cigarette, you will get that. And if you're using UV light in any process in your pipeline, UV light, if you have rubber materials exposed to UV light, you might also get some ethylene produced. Now, how much it is, it depends on your operations, and this is why you need to know what's going on, how much ethylene you have. You want to manage ethylene so you can end up getting the result you want in your product, right? The next thing you need to do is learn how to measure ethylene. And I hope throughout this workshop you get the opportunity to learn about different technologies and different ways to approach that are suitable to your different operations. But I wanted to bring it up because some people might avoid this because they say, well, this is too expensive, I don't want to do it. But at the end, the payoff of understanding ethylene levels in different parts of your operation might be more cost effective. So it actually is a routine, and possibly many of you already have gotten exposed to these type of measurements. I put some here and will depend on the type of operation, but commonly used methods are ethylene detection tubes. These ones here that you can see in this part of the slide are the Kitagawa ethylene detection tubes. And these are easy to use, but are not as accurate as other methods. So they're cost effective, but might not be that precise. And then you have other proprietary analyzers. I bet you're gonna get exposed to some of these technologies at the end of the course. And you have some things that we use mostly for research. You can use a gas chromatograph, or you can also go fancy and use something like a photoacoustic detector. So what will you use will depend on your needs, and you have to balance that between how accurate you want to be and the price, or how much money you want to invest on these types of methods. So here I put in a slide about chromatograph and gas chromatography that we use a lot in the lab. And basically what you do is that you inject a sample throughout this system, and after it goes through this capillary column and goes through a detector, you get this in a screen, could be either in a computer or in a piece of paper, and you see these peaks, and you basically, in the case of ethylene, you can detect based on time when your ethylene peak will show up, and the concentration of ethylene will depend on the size of this peak and on the area below the peak. So as I said, you have ethylene as a friend, in which will render benefits for your product. It can basically induce faster or more uniform ripening when we talk about climacteric fruit. I will go more in depth on that in a little bit. It can also promote ripening events in fruit that are non-climacteric, for instance, the biosynthesis of pigments or degradation of chlorophyll. And using ethylene to improve quality is not something new. It has been used many centuries or years ago by even ancient Egyptians that were able to use either incense or smoke to ripen their products. So as I told you, they didn't know it was ethylene itself, but causing an incomplete combustion of certain products, let's say like incense, you can have the release of ethylene, and if you have an enclosed chamber and you have ethylene present, you're able to get these effects of ethylene. So now this is back in the time. We are understanding more how things work. We know is ethylene the driver for climacteric fruit ripening, and we know that there's many ways that we can control it. So why we have what we said, climacteric and non-climacteric ripening, and how does this relate to ethylene itself? Well, as probably in the first or second presentation you heard about climacteric and non-climacteric fruit, you probably talk about it in your companies or in your institutions. But what does this really mean? So climacteric fruit are fruit that can be harvested when mature, physiologically mature but not yet ripened, and you can ripen it using exogenous ethylene. And of course, climacteric fruit depend on ethylene, and you will see the rate of respiration will increase throughout ripening, and also you'll see an endogenous or plant producing its own ethylene. So you'll see a burst of ethylene. Now, non-climacteric fruit are generally, they need to be ripened. If you don't harvest them ripe or close to ripe, you may never get quality on that fruit. So even if you expose it to ethylene, nothing really happened. Ethylene may change a bit in that fruit. Maybe some production might be seen, but really low, and it doesn't really have a big effect, as I was saying. So we know this for a long time, and that's why we have differences in how we manage fruit in different scenarios. So here is not an exhaustive, but a complete list of non-climacteric and climacteric fruit. And when we talked about climacteric fruit, as I was saying, you have, for instance, an unripe fruit that undergoes ripening, and you get all these nice things, red color or coloration, aroma, flavors, sugars. You have a better texture because of soluble pectins and so on. And you have a reduction in things that are really linked to unripe fruit or unripe fruit, like green color or acidity goes down, starch goes down. So these are linked to quality. And as these things go up, you can see ethylene. Generally, there's a burst in ethylene, and respiration is just before you will see an increase in CO2. So how can we compare climacteric to non-climacteric ripening? So here is an example. So non-climacteric ripening is all these dashed lines. So ethylene will not really be produced. Respiration really goes down. But the fruit is growing and is ripening. It's just, it doesn't require any of these two processes to happen. Now, for practical uses, we say this is climacteric and this is non-climacteric. It's easy, right, to separate things in two categories. But nothing in life is two things, right? There's always sort of a range of things. In this case, even though we know examples of fruit that are climacteric, like a banana, there's always the possibility of fruit that end up a little bit in the middle. They might need some ethylene, but are not totally depending on ethylene, and might be depending on other hormones. So here is an example. So we have a gradient of fruit. These are what we call climacteric, and these are some non-climacteric fruit. And in the middle we have other fruit that strawberries are generally called non-climacteric, and tomatoes and apples are climacteric. But the dependence on ethylene may be different. So the amount or the dose of ethylene that you need for each of them might be different. And if you understand that, and other players may be important for ripening, maybe other hormones or other factors. So understanding that not everything is ethylene is quite important for you. And so don't always think climacteric one way, non-climacteric. It's always a moving gradient, and think about that. Okay, so as I was saying, depending on the fruit and the stage and the ripening program, you will need certain conditions or certain parameters for ripen that fruit. So ripening fruit, usually using exogenous ethylene, is effective only when the fruit is capable to perceive ethylene, when it's physiologically mature. Many commodities are generally treated with ethylene. Some of them include, here in California, avocados, banana, tomatoes, mangoes, and then in other places you will see papaya, nectarines, melons, and others that are generally treated with ethylene. So the process, in the case of banana, you have a more in-depth presentation, but you probably will need some pre-cool step or ethylene exposure, and you also will have some ventilation and post-cool storing. Now, bananas are chilling sensitive, so you will have to also understand that the cold conditions or chilling conditions may vary between different commodities. So an example on how much ethylene, how much time exposed to ethylene some commodities need. This is what generally is used in ethylene ripening chambers. It's 100 ppm. I told you that plants can even perceive down to 0.1 ppm, but we always kind of pushed our boundaries. So 100 ppm ethylene to have a more uniform response. So avocado needs about 8 to 48 hours. Tomato will need about 24 to 72 hours. And I told you, depending on the commodity, the temperature that you will need for that ripening to happen may vary. So here is an example in Celsius and Fahrenheit for you to have an idea. So, considerations for ethylene treatments. I told you the threshold concentration has to be 0.1 ppm. This is really low. People always go above that. But generally, responses plateau at 10 ppm. So we expose them to 100, but maybe you will not gain that much of a difference between 10 and 100 ppm. Now, for practical purposes, sometimes it's easier to manage higher concentrations of ethylene, and that's why you expose to 100. That getting it to a 10 ppm concentration might be tricky when you are setting up a ripening room. So that's why generally is used in that concentration. And you will also need to consider that there's going to be an increase in respiration due to ethylene, so your commodities will start respiring. What does that mean? Metabolism will kick, and what happens when things start to metabolize faster? They start dying faster, they start losing shelf life faster. So yes, as we had a question before, you ripen things, but as soon as things are starting to ripen, you're starting also to lead to that part of the development that is going to death, to senescence, to dying of the tissues. So you have a window of opportunity from the ripening to selling your product before things start going sour. And as I said, you need to understand temperature, and you need to understand the gases, if there's CO2 presence, because a lot of CO2 inhibits ethylene responses. So if you're respiring, what are you producing? CO2, right? So if you put too much ethylene, you get CO2 produced by your fruit, and if you don't control the accumulation of CO2 in that chamber, you basically are inhibiting it. So you end up not getting the result you want. So when you design a ripening room, you have to have those considerations. And this is an example of a ripening room. So you need to know your ethylene concentration. Yeah, please don't go over 100 ppm. It's not necessary, as I mentioned. The temperature you need to use is the lowest possible for your commodity. So because you want to keep that metabolism a little bit under check, so don't go too high. The humidity needs to be also accurate, appropriate for the commodity. You want to have high humidity so you don't have dehydration because of the rise in respiration that you may have. Depending on the commodity, you will have to have certain duration of exposure to ethylene. Again, the air circulation is critical, so you have an uniform distribution of the ethylene, but also you don't get enough CO2 accumulating in certain parts. And sometimes you might need even a CO2 scrubber or a sort of a good ventilation system, so you remove that CO2 that is accumulating. So here is an example of a diagram of a ripening room. And people, depending on where they're from, they might use ethylene cylinders or generators. We know that ethylene is flammable and is also can be dangerous. So most of the times cylinders have been changed for generators to avoid that and to have a better control on the ethylene production. So I know you will get some examples. I saw one of these ethylene generators in the other room, so you will get to see it if you have never been exposed to those. And you basically, in these generators, what you have is ethanol and a copper catalyst they are both exposed to heat, and they release ethylene in an accurate manner. And besides producing the ethylene, you might also need a control, so you make sure that you don't go higher than those 100 ppm or lower, that you will not get exposure to it. Just to finish my presentation, I told you that ripening can be our friend if we know how to manage it, but also can give us some disadvantages if we don't control it. So sometimes your fruit is already ripe and you don't want it to go mushy or overripe, so you need to control the ethylene. You need to compartmentalize where you have ripening happening from when your fruit is ready. You want to send it. You don't want that to be exposed further to ethylene. That's an example. Because then you will start getting death. You're going to start getting senescence, and you don't want that. In certain products that might be shipped together, if you have ethylene present, you might have chlorophyll loss. You might also get sprouting, as I show you in potatoes. You might promote abscission in certain commodities, but this is mostly in the field. And you stimulate respiration, what I told you, so you get CO2 produced. You might get wilting, and also if there's too much ethylene, it doesn't really help you to prevent disease, but it instead will cause decay. So things that are really sensitive to ethylene are generally vegetables. There's a list of different commodities and what happens if they are exposed to high doses of ethylene or for extended periods of time. So that's what I show you. You get yellowing, you may get some decay or browning of the tissues. One thing that is really sensitive and you do not want to have closer to ethylene are ornamentals, basically flowers. So never ship flowers with apples. That's not a good idea. Flowers will lose quality pretty rapidly. Even potted plants will have abscission, which I mentioned. So you might lose your flowers, you might lose the quality of your ornamental. So think about that. You need to manage ethylene differently. So as I said, most of the damaging effects of ethylene are associating with the induction of senescence. So that's the end of the life of an organ or a plant product. It causes the breakdown of chlorophyll, what gives the green color to certain commodities, which give the appearance of fresh or ready to eat for certain products. When you find a broccoli that looks like this, you're not, this is way too old, I'm not gonna eat it, it's not fresh enough, because it already went through all this degradation of chlorophyll and dying senescence processes. So one thing that is important is that the effect of ethylene is accumulative. So it depends on how much time things get exposed, you're gonna have that either favorable or detrimental effect. So if you put together an apple and a broccoli for a few minutes in your shopping cart when you're in the supermarket, maybe nothing will happen, right? Because the time of exposure is small, the amount of air in the environment is small. But if you're putting a container full of broccoli and full of fruit, you might see this after you shipped your container somewhere, right? So these things are important. How long are things stored together, are put together, is necessarily to be accounted for. So if you're in a distribution facility where things move fast, in an hour or so things are changing places, you might not have to be as critical about your ethylene. But if it's something that you're going to store for a while, you might need to be able to control and measure ethylene. So that being said, I'm opening basically the ground for the next presentation, which will talk about management of ethylene in post-harvest. So we will have Angelos discussing this in a little bit, and he will talk about how can you avoid ethylene responses that are undesirable. So you have any questions for me, and I'll be happy to take them.